Hi, I'm Marina Garrett. I'm an assistant investigator here at the Institute. This is Pete Groblewski. He's the senior manager of the neurosurgery and behavior team. And as the co-leads of the Visual Behavior Project, we're really grateful to be representing the very large team of people that's involved in this work, many talented scientists, engineers, and technicians. So it's really a privilege to work with a group of such dedicated people. I need the clicker. Here we go. So we're excited to share some recent results with you today, looking at one of our key questions about how sensory coding is influenced by experience, expectation, and task engagement during a visual change detection task in mice. So you'll hear from several members of the team about results with different data modalities, but using a shared behavioral task. So first, I will tell you about how population activity measured with 2-photon calcium imaging is modulated by experience and task engagement. Then Nick will go into a little more detail about an exciting result showing that VIP inhibitory cells have a rapid experience-dependent shift in dynamics. Then Farzaneh will show you how our multiplane 2-photon imaging platform can be used to study how network interactions differ following expected and unexpected events. And then finally, Sam will tell you about some results with Neuropixels recordings showing that the correlation between neural activity and task performance is area dependent and increases along the visual hierarchy. So before we get into all the results, Pete's going to tell you about the data collection pipelines that we've built and describe the image change detection task that's shared across the pipelines.
Thanks. So in order to address these questions, we've built these Allen brain observatories, which are high throughput, large scale, in vivo pipelines, and they consist of a series of experimental data collection as well as data processing steps, each of which is performed by a team of trained technicians in a very standardized manner. I should note that all of the data sets that we collect in our brain observatories are made freely available for continued analysis at brainmap.org. So for the Visual Behavior Project, we've actually developed 3 pipelines. We have our single and multiplane 2-photon calcium imaging pipelines, which are currently in production. And we also have our Neuropixels pipeline, which is coming into production in the next few months. So the single plane 2P pipeline allows us to image from genetically defined subpopulations of both excitatory and inhibitory cells. We can do this across many days. And our multiplane platform allows us to make interareal as well as interdepth comparisons within these same subpopulations. And lastly, our Neuropixels allows us to make similar interareal and interdepth comparisons, but with much higher temporal resolution. So all 3 of these pipelines share the same upstream standardized workflow, including behavior, in which we train mice on an image change detection task. So in this task, mice learn to emit a behavioral response when the identity of a flashed image changes, while withholding a response when the image of identity of the image does not change. So we've trained hundreds of mice on this task in our pipeline, and they readily learn the task in a matter of 2 to 4 weeks. And here's an example of one of our performers. There you go. Okay, so as you can see, the mouse is able to withhold its licking while the identity of the image shown on the screen in the background doesn't change. But as soon as that identity changes, the animal emits a single lick on that spout, and that lick triggers a water reward, and then the animal is free to consume it and move on to the next trial. So these animals can perform hundreds of back-to-back trials on a given imaging session, which makes it really well suited for our physiology experiments. So with that, I'll pass it back to Marina, who will tell you how we're leveraging this behavior for our first visual behavior pipeline project. You can just let it watch.
I'll let it go. Okay, so to get at our questions about experience, expectation, and task engagement, we've designed an experiment that starts with mice being trained in the image change detection task that Pete just showed you, and they are trained with a specific set of 8 natural scene images. And these images become highly familiar over training. So here, each of these red blocks indicates one 1-hour behavioral session with this set of images. And so they see them, tens of thousands of times during training. And then during the imaging phase of the experiment, they see, again, that same set of 8 images in 3 different 1-hour sessions on separate days. So there are 2 active behavior sessions, this one here, A1 and A3. This is the mouse doing the task just as they were doing during training, with an interleaved passive viewing session, A2, where the mice get their daily water before the session, and so they're sated and they're no longer motivated. And then the stimulus is presented with the lick spout retracted, so the mice cannot engage with the stimulus and earn rewards. And this allows us to compare the activity of neurons in visual cortex when the animal is actively performing this behavior and seeking rewards versus a disengaged condition on these passive days. So in addition to the familiar images, image set A here, we also show the mice a novel set of images that they have not seen before the 2-photon microscope, again in 3 separate sessions, 2 active behavior, B1 and B3, with a passive session in the middle. And within each of these sessions, as Pete just described, the sequence of image presentations is highly regular. So they see a 250 millisecond image flash followed by 500 milliseconds of gray screen. And during the behavioral training, that sequence is totally uninterrupted. And so they have extensive experience with this predictable timing of the image flashes. However, during the 2-photon imaging sessions, we actually omit 5% of all non-change flashes. So there's changes that happen when the mice need to emit their behavioral response, but there's also many non-change flashes that happen during this continuous stream of images. And so what we do is omit 5% of those non-change flashes. And this allows us to ask how neurons in the visual cortex react when an expected event is omitted from this predictable sequence. And so this feature of the task will become important for the results Nick and Farzaneh will show in a few slides. So one of the first questions we asked with this experimental design was whether the mice would perform the task when they see novel images. And so here I'm showing a measure of behavioral performance, D prime, for the last session here with the familiar image set, A3, and the first session with the novel image set, B1. And you can see that on average, their performance is very similar. So essentially, they rapidly generalize this behavior to a novel set of images, which tells us that they've learned the task rules, they know they have to respond to changes, and they're not disrupted by this novel stimulus being presented. Another question was whether the passive viewing sessions interleaved between active behavior sessions would disrupt their behavior. Going through a session where they're not receiving rewards in the typical way, whether that would affect anything. And we see that at least on average, their performance is pretty stable before and after these passive sessions, both for the familiar image set and the novel image set. Given that behavior is roughly similar on average, are there any differences in the physiology of neurons and visual cortex across these different session types? And we do find differences. So here I'm going to walk you through this plot where now I'm showing the average population response across all excitatory cells in layer 2-3 of E1 just during this first session with the familiar image set that they were trained on. And this is triggered on a change time. So there was a change in image identity from something to something else, and this is average across all images. And you can just see that there's a clear stimulus-evoked response at the time of this change. Then we can go to the passive session, A2, and you see a reduction in the strength of the population activity during this passive session. So this is suggesting that there's this enhancement with task engagement compared to disengaged condition during the passive session. Then we can again look at this A3 session, and we see that actually the strength of the response recovers back to the level of the A1, the first active behavior session. And so this is where this interleaved design is actually really important to show that this difference here is actually a result of the engagement or motivation state of the animal and not something that's maybe just changing over time. So the fact that we can see this recover back to the initial level is useful. Okay, so then we can ask, what happens when you show novel images? So we saw the behavior was roughly similar, but when you look at the population response for the first session of the novel image set, you get a very drastic difference in the magnitude of the population response. It's much, much larger. So there's this effect of novelty here. And then we can look at the second session with this new novel image set. This is again a passive session where the mice are disengaged from the task, and actually the strength of activity goes down, about to the same level as the passive session for the familiar image set that they've seen many times. And then finally, the very last session, active behavior session with the novel image set, actually has this intermediate level of activity. So it's reduced relative to the first exposure to this novel image set, but it is still higher than the disengaged case. So I think this is really cool. You can see both effects of task engagement and experience within the same plot. So the active-passive differences, and then effects of experience comparing something that's highly familiar with something that's very novel, and comparing something that's, the very first day where it's novel compared to after a couple days of exposure, and you see a difference even there. So we can also look at layer 5 excitatory neurons. So here showing all 3 familiar image set sessions, and we see this similar trend where the 2 active behavior sessions, A1 and A3, have a larger population response magnitude compared to the passive day here. And again, with the novel image set, you see a similar trend as in layer 2-3 cells, where the passive session is the lowest, the first exposure to the novel image set is the highest, and the third exposure is intermediate state. So one of the most exciting things that we saw is when we looked at the activity of VIP inhibitory neurons. And so these are cells that provide local inhibition, primarily in layer 2-3, targeting many populations, but particularly somatostatin neurons, which disinhibit the apical dendrites of pyramidal cells. So while this is an active area of investigation, the current idea is that the effect of VIP cells should be to disinhibit the local pyramidal cell population. So what do VIP cells look like in these different conditions? Here, looking across all of the familiar image set cases, we see very little response. If anything, there's this kind of increase in activity prior to the time of the image onset, and then kind of a decrease afterwards, so maybe some kind of interstimulus ramping behavior. And where it becomes striking is when you look at the first session of the novel image set. Now you get very different response profile in VIP cells, where they have a very large sensory evoked response. It's clearly active after the stimulus onset, not before. And this profile actually changes very rapidly. So by the second session with the novel image set, this passive B2 session, and also looking at B3, you get a big reduction in the response magnitude. And also you see this shift back to these kind of pre-stimulus ramping dynamics. So this is, pretty striking. And I actually will pass it off to Nick to tell you a little more about how these dynamics change within a session, and also how VIP cells respond when something expected is omitted from the sequence.
Hi, everybody. I'm Nick. I'm a scientist here doing analysis as part of the visual behavior team. And I've been looking at the changes that we see in VIP responses by taking advantage of the omitted stimuli that Marina mentioned before. So just to bring you back, during training, the images are presented with a very regular sequence, 250 millisecond image presentations with 500 milliseconds of gray in between. But when we move animals from behavior training to imaging, we begin to randomly omit 5% of non-change flashes. And during this time, we can see what happens to things like that pre-stimulus ramping activity in VIP neurons if you don't present a stimulus. So I'm going to present responses of VIP neurons that have been aligned to the time when the stimulus would have come on, but in cases where that stimulus was omitted and not presented. On the y-axis here, we have a bunch of different VIP neurons that we've recorded. The x-axis here is that time relative to omission, and then color here indicates the activity of the neuron measured as the change in calcium fluorescence. So we've also got a population average trace, just averaged across all of these neurons down here. And what we can see in this session, which is the first session with that familiar image set that they've been trained with, is that the VIP neurons simply continue to ramp up after the omission until the time of the next stimulus, and then their activity decays back down. So here's activity across the next two presentations with that familiar image set. And we can see that both within session, there's differences in the strength of this response across neurons, and then across these three sessions, there is some difference in the strength of that ramping response. But what's really striking, again, is when you switch to the familiar image set, to the novel image set, that first day with the new images B, we see that these VIP neurons switch to actually reliably responding after image flashes. And on average, across this first session, being very quiet during this period where there's the omitted stimulus. However, the very next day, with that new image set, we can see the recovery of this ramping activity during the omitted period, which then decays following that next image presentation. And that actually becomes even stronger on the third day. So there's a couple of questions that we've been addressing. One is, how do responses of individual neurons that we can track across all the days of our experiment change under these different conditions? And the second thing we want to look at is what's going on during this first day with images B. How quickly do we see responses start to change? So to address the first question, I've got here just one example VIP neuron that we're able to track across our entire experiment. In the top row, we've got the image of the cell body of the neuron. And then in the middle row here, we have the activity of this neuron aligned to the omissions. And then in the bottom here, we have the activity on each omission in the trial. In the session. So we can see that for the sessions with the familiar image set, this particular neuron ramps up following that omission. But then when you switch to the novel image set, this neuron also changes its response to displaying this stimulus evoked response and being very quiet after omission. And then we can see some ramping activity recover on the next day with B, and then it's strongly ramping again by that third day with the images B. However, there is some variability across the session, and so that leads me back to my second question, which is what's going on during the first session with images B. And so here, I've taken that the average population response of VIP neurons across that first session, and I've split it up into 6 blocks, where the blocks early in the session are plotted in these purple colors, and the blocks later in the session are plotted in these orange colors. And what we can see is that at the beginning of the session, the population is very quiet following the omission, and then displays the large stimulus evoked response to the next flash. But that across this first session, this 1 hour of experience with images B, we see the development of this ramping activity that follows the omission. And what's interesting is that we also see across this first session a reduction in the stimulus evoked component following flashes. So we see this trade-off in the flash and the emission activity over time in the B1 session. So now I'm going to pass it off to Farzaneh, who's going to tell you how different areas and layers interact following these expected and unexpected events.
Thanks, Sam. Hi, I'm Farzaneh. I'm a scientist at the Institute, and I'm going to talk about interaction of cortical planes during expected and unexpected events. So the data that Marina and Nick showed you were about the response of excitatory neurons and VIP cells to flashes and omissions, but that was all done in V1 and mostly in layer 2-3. But as Nick mentioned, of course, all these brain areas and cortical layers are interconnected, and to get a proper understanding of their function, we have to look at their activity at the same time. For that, we leverage the data from multiscopes. Multiscopes are advanced 2-photon microscopes that we have developed at the Institute, and they allow imaging 8 cortical planes simultaneously. We picked 2 areas, V1 and LM. LM is a secondary visual area that is highly connected to V1, and also 4 cortical depth that roughly correspond to layers 1, 2, 3, 4, and perhaps superficial 5. To study interaction across cortical planes, and particularly because these planes are imaged simultaneously, we can properly address that question. As a first step measure, I use pairwise Spearman correlation coefficients. For that, I took pairs of neurons, one in each plane or within the same plane, and I took the activities that follow the flash. And I compared the correlation coefficient between the activity of neurons that are evoked by the flash. I did the same analysis now for the activities that follow the omission. And the idea here is that does the activity of neurons similarly covary after flashes which are expected versus omissions which are unexpected? I did all of this analysis on A sessions which are familiar. So let's see how different cell types interact after flashes and omissions. When we look at excitatory neurons, we see that correlation among excitatory neurons increases much more after flashes versus omissions. And this is true for V1 particularly, also for neurons with an LM and between V1 and LM. If we look at the inhibitory SST cells, we see a similar pattern. They become highly correlated after flashes and not much after omissions. Just how Nick showed you in terms of their activities, we see that VIP neurons, in terms of their correlations, show a very distinct pattern compared to excitatory and SST cells. They are highly correlated after omissions, but not after the flashes. So what we conclude here is that after expected stimuli flashes, we see strong coactivation of the excitatory and SST cells in V1 and LM. However, after unexpected omissions of the stimulus, we see strong coactivation of the VIP neurons. But how about layers? Is there a difference in how different cortical layers interact? For that, I repeated the same analysis, except now I'm taking neurons from each layer, and I take pairs of layers, and I compute correlations there. Here on the x-axis, I'm showing the depth in V1, and y-axis is the depth in LM. The brightness of the color is indicating the strength of the correlations for the excitatory cells. So overall, we see that correlations are higher for flash-evoked responses compared to emissions. And then, interestingly, we see that the strongest correlations are happening between neurons that lie in deep layers of V1 with LM. This is actually in line with the existing evidence that says some sensory inputs are relayed from thalamus to layer 4 and 5 of the visual cortex. So they're receiving this common sensory input, and as a result, they're co-activated. But how about SST inhibitory neurons? Here, again, correlations are overall higher after flashes compared to emissions. And again, we see deeper layers of V1 seem to be more correlated with the LM. When we look at VIP, again, there is a striking different pattern of coactivation. No coactivation after flashes, high coactivation after omissions. And it seems that the current data is suggesting that unlike excitatory and SST cells, which were more coactive in deeper layers, here we see more coactivation in superficial layers of V1. We have to confirm this with more data, but if it is true, it's actually a very exciting result, because the current evidence suggests that feedback input that carries prediction signals is more related to superficial layers of the cortex. So what I have shown you is that flashes and omissions evoke very distinct patterns of activity in the cortex. Flashes are more strongly represented in excitatory and SST cells, and particularly in deeper depth, and omissions are more strongly represented by VIP neurons and in superficial layers. Now I'm going to pass it on to Sam, and he'll talk about recordings from more areas and at better time resolution.
All right, so calcium imaging, as you've seen, is great for following identified cell types over many days. The advantage of the Neuropixels probes, of course, is that we get much higher temporal resolution as we record from many areas simultaneously. And another advantage is that we can also stick our probes down deeper in the brain, as Nick showed, and record from subcortical areas also. So here's an example recording from 6 probes simultaneously, about 700 neurons, across various areas, where each row here is a neuron, and each little dot is a one action potential or spike of a neuron. And this is for one trial of the change detection task. And so you can see there's lots of activity associated with the image flashes and the change, and with the animal's motor response in response to the change. And a fundamental question is, when relative to the change, is activity important, and across which areas? So we'd like to start addressing
I'm told to tell you to stand closer.
that. Okay, so the first thing we look at to ask like when activity might be important is just see how long it takes the animal to respond to change. And so when we look at the animal's lick, which he uses to get his reward, they first start licking at about 250 milliseconds after the change. However, the animals are running during the task, and before they start licking, they invariably slow down. So if you look at the running speed, you can see that the mouse is already starting to slow down, is running about 150 milliseconds after the change. So that suggests that the animal is just using sort of this first wave of cortical activity to detect change. We've recently started some experiments to potentially narrow down or refine this temporal window, where we use optogenetic inactivation of visual cortex, either starting at the time of the image change or at various time points after the change. What we see is when we inactivate visual cortex at the time of the image change, that there is a very low rate of responding, similar to the animal's false alarm rate. But that as the cortical inactivation begins at later and later time points after the change, the animal is then able to respond. And it looks like somewhere between 100 and 200 milliseconds of activity is important for the animal's detection of change. So this sort of preliminary suggests that the animal is using visual cortex almost all the way up to the time of his behavioral response during this task. Okay, so that addresses the question of when activity is important. We'd also like to narrow down which of these areas are most important for how the animal detects change. And to do this, we're going to take advantage of the fact that there's variability in the animal's behavior. Some image changes he detects, and other ones he misses. And so the way we're going to look at which areas might be most important is we're going to train a decoder to distinguish snippets of activity associated with either the pre-change flash or the change image flash, and tell us which of those population activities are associated with change. We can then test the decoder with activity for changes that the decoder was never trained with. And we see results like this. And so you'll notice in this example that the performance or accuracy of the mouse and the two decoders, either the decoder based on V1 activity or the decoder based on AM activity, is exactly the same. They both detect 2 of the changes and miss 2 of the image changes. But whereas the trials that the mouse and the AM decoder detect or miss change are exactly the same, this V1 decoder is detecting something that the mouse missed and missing something that the mouse detected. And so it's this relationship between the decoder's prediction and the mouse's trial-to-trial variability that we're interested in. So when we do this for hundreds of trials over many mice, here's what we see. We see that the accuracy that we're able to decode change is similar regardless of where we use activity from, which brain regions. But the correlation between mouse behavior and the decoder's prediction increases as we move up the visual hierarchy, and particularly in higher order areas such as AM. So this suggests that activity in AM, for example, might be particularly relevant for how the animal detects change. So in summary, today, as a team, we've showed you that population activity is modulated by experience and engagement, that VIP neuron and dynamics shift rapidly with experience, that network interactions differ following expected and unexpected events, and that the correlation between neural activity and task performance is area dependent. Thanks for listening, and we're happy to take questions.
All right, we have time for a few questions, so if you raise your hand, we can get microphones to you.
Hi, here. I have a question for the VIP unexpected stimulus activity. Do you need a task for that? I mean, because this is not task related, I guess.
Sure. So I'm not sure actually if you need the animal to be engaged or not to uncover that. Definitely the switch from a familiar image set that they've experienced a ton of times to the novel image set. There's big differences in that response. So maybe all you need is some experience-dependent switch like that.
I think the answer is we don't know, right? We could try to run the same exact stimulus paradigm with the right timing and familiar, novel, and all that in a passive case where the mouse never gets any rewards and see if you still see the same effect. So it's a really good question.
I'm interested to learn a little more about what happens during the novel images. So you claimed that the behavior generalizes. So, but for that you would have to show that this behavior happens the very first time that the mouse sees the novel stimulus. So I'm wondering what happens at the behavior when you switch to the new stimulus set.
Yeah, that's a good question because we're showing behavior on average across the entire 1 hour session. So is there a difference in their accuracy, response latency, or something like that across the session? We're looking into that right now. So giving precise quantitative details is challenging, but just overall, it looks like they're generalizing very rapidly. So if there is any difference, it's within the first 10 or 20 trials. But after that, their hit rate and their response latency is very similar to the familiar images. So I think there may be some changes at the very beginning that we're still looking into the details of that, but it's fast.
I'd like to ask about the relationship between the sort of surprise modulation of VIP activity and some of the classic older results that show that VIP is co-modulated with running speed. And do you think these are part and parcel of the same sort of quantity, or that there are multiple populations of VIP cells that are serving different purposes, or what? Keen to hear your thoughts.
Well, the idea of whether there's multiple subpopulations is super interesting. That's another thing we're starting to kind of look into and see if there's differences within the VIP group. But in terms of VIP and running, there's actually an interesting result that we didn't show today that the mice kind of lock their running behavior to the image flashes. They slow down a little bit right before each one, which is actually the opposite direction of the VIP signal, which is maybe not expected from some other results showing that they covary. So yeah, that's another thing we need to follow up on and look into. But yeah, they do seem to show the running behavior and the VIP ramping pre-stimulus are in the opposite direction effectively.
Any more questions? All right, if no more questions, let's thank our speakers again for a great talk.